Welcome back, everybody. So we will be proceeding from the inheritance of traits. Mendel's law of segregation, it's not confusing. It's very straightforward. The law of segregation says that two alleles for each trait separate during meiosis. So during fertilization, the two alleles for that trait will reunite again. So let's say, for example, as we can see here, we have two traits, which is this and this. So these are the two alleles for a trait, which is a yellow, which is color. So these two alleles code for yellow color. So when a gamete is about to form, they would separate into two individual allele. So this is what the law of segregation says. So it says the two alleles would, for each trait, will separate this way during meiosis. So during fertilization, the two alleles for that trait would reunite again. So the heterozygous organisms are called a hybrid. So the meaning is once they separate, so only one of it would be used for fertilization, and the other one, which has got this also, would separate into this, and only one of it will be used to proceed to fertilization. So the fertilization process would now have this and what and this. So that would give you this, which is now a hybrid. So we call that a F1 hybrid because it's a combination of two different alleles. So this is the reason it says in the flower, in the pollen grains flower, so in the pea flower, so the pollen grains will be used. So we use the pollen grain and the what and the anther produces a pollen grain, and we put it on the stigma of another one. So that's the reason it says one pollen grain and one egg. So then it fused together, it forms a zygote, which is a hybrid, where it develops a a seed, which is a yellow, because there is one dominant allele for the color yellow in it. So this is the reason why we have it that way. So the next one is the law of independent assortment. So we have two main laws here. So we talked about the first one, the law of segregation. That is a law that says that the two alleles for each trait would separate, as you've seen here, during meiosis. All right, so what about the law of independent assortment? The law of independent assortment states that a random distribution of alleles occurs during gamete formation. So when you have set of alleles, there will be a random assortment. There is no kind of a specific way in which they happen. So genes on separate chromosomes sort independently during meiosis. So each allele combination is equally likely to occur. So what does that mean? You could see we have some alleles here. So we have heterozygous yellow, and we have heterozygous green color. So these are the four alleles we have. So these are the possible combinations we can have from them when the when the gamete formation is happening. So this one can form with this one. So this one. So mind you, you must not cross two Ys, and you must never cross two Rs. So it's going to be one Y with one R and one Y with one R. So when you do this, you will have these results here. So the next one is the Punnett square. So this is the Punnett square. So on a Punnett square, so the monohybrid cross is in the Punnett square when you have a monohybrid cross. When you want to cross organism which are monohybrid, So, so you have a monohybrid here, and you have another monohybrid. So Punnett square helps to keep track of possible genotypes involved in a cross. So what do we see here? We have this and we have this. So then you would have one gamete from this and you have one gamete from this. So when these two gametes come together, they produce a yellow pea because there is one dominant yellow pea here and there's one a recessive green pea. So then it's gonna give you a combination which is gonna be a yellow pea. So a cross that helps hybrid to form a single trait is called monohybrid cross. So the cross that helps to form a single trait is called a monohybrid. So what is the word monohybrid? The meaning is you're actually crossing one trait at a point in time. Now, for example, we want to make a cross to get the color distribution. So what do? We have a yellow pea, which is homozygous dominant. And we have another one, which is homozygous recessive. So what do? We take a gamete from this, and we take a gamete from this. So these two gametes fused together, they give you heterozygous F1 generation. So because this is a dominant and this is recessive, it's going to give you a yellow pea. It's going to give you a yellow pea, as you see here. So, but sometimes when they self-pollinate, when this hybrid self-pollinates, that is to say when there's a cross between it, so what would you see? So what you would say is when you have this, 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 and this. You can use a Punnett square, it's pretty easier. So that would give you this, 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 and this, this, and this. So you're gonna have, sorry, this one with this. So um, you're gonna have one homozygous dominant, two heterozygous dominant, and one homozygous recessive. So this is what you're gonna have. So but the phenotype would be yellow for this, yellow for this two will be yellow, and this will be green. So that's why you have ratio 3:1 as a phenotype. So a monohybrid cross, another example is a freckless and freckless missing. When you have a parent, so one parent is having freckles and the other one doesn't have a freckles. So the number of squares is determined by the number of different types of alleles. So you have this and this produced by each parent. In this cross, the genotypic ratio is ratio 1 to 2 to 1. So the phenotypic ratio is 3:1. So you would see if you cross this properly, you will notice that these two parents are heterozygous. So this is a parent who is heterozygous to freckles, and this one is heterozygous to freckles. So the meaning is if you have one dominant, you have one dominant, and you have one recessive. Both parents have heterozygous. So when you cross two heterozygous parents, it's going to give you one homozygous dominant, two heterozygous dominant, and one homozygous recessive. So if you want to know how to find a genotypic ratio, what you would do is pretty easy. You will count. This one is different, it's 1. These two are the same, it's 2. This one is different, it's 1. So the genotypic ratio will be 1 to ratio 2 to ratio 1. That is to say, F of homozygous dominant F is 1 to ratio heterozygous dominant to ratio homozygous recessive. So that is the genotypic ratio. But when we ask for phenotypic ratio, What we're talking about is we want you to, based on what you see, based on the number of kids that have freckles, you will count this one would have, this one would have, this one would have, but this wouldn't have. So it's going to give you ratio 3 to 1 to 1. That's the phenotype. Guys, please understand these types of calculations very well. All right, so now where we have, mind you, we're talking about monohybrid, where you're considering only one trait at a time. But sometimes we consider multiple traits at a time. Now we want to consider two traits at the same time. So the dihybrid cross, the simultaneous inheritance of two or more traits in the same plant is a dihybrid cross. So you want to consider two or more traits in the same plant at the same time. So there is di is meaning more than one. So four types of alleles form the male gamete and four types of alleles form the female gamete. So you have four alleles for the male, which is the parent. So the parent is having homozygous dominant for color. Excuse me. So what I'm saying is, if you have these two, you have homozygous dominant color, homozygous dominant smoothness. So it's smooth if it's uppercase R. So and this guy is got all of homozygous recessive color, which is green, and homozygous recessive texture, which is wrinkled. So you're considering two things. You're considering being green and wrinkled. This is being yellow and smooth. So you should be able to use the genetic code to tell us the phenotype. So what do we have? We would choose one of these to form a gamete, one Y and one R. You're going to choose one Y and one R. So once you have it, then you will cross them together. Someone might ask you, oh, how did you get these? Why is it so complex? It's not complex. It's just for you to cross them properly. So what you can do first is you would put your Y and R on a Punnett square. Then you put your Y and R here on the second side of the Punnett square. So once you do it like this, then you will have YI, YR, YR, and no, this is a mistake here. So you will do it like this. It's easier. So what we want to consider is we want to consider a Y. We want to consider a Y, a R, and a Y, a R. So all you're gonna do is you put your Y, and your R, and you put your Y, your R, Y, and R, and R. So once you cross this, so it's gonna give you YR, 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 YR. So you will have this. So once you have this, so this will produce, this will be the main yardstick for your crossing. So this is the male, this is the female. So the male here, the female there. So some people might say, oh, this is very confusing. It's not confusing. It's pretty easy. Let me do it again. So we have, I think you understood how we took one Y from here, one R, one Y, one R. So he crossed it. So I bring the Y, the R. So it's just like you have this, the same thing I did for you before. Let me remove this and put it again. So we have this Y, R, and Y, R. So once you do the cross properly, so this will give you YR. You do it again with this, it gives you YR. So then you do this one, with this, it gives you YR. And with this, it gives you YR. So this is how we got this. So you can either line them up like this or use a Punnett square. Whichever you find easier for yourself, it's okay. So the Punnett square, you would have on the Punnett square, you would have Y and Y, R and R. Sorry for me writing on the, I'm just trying to show you because some students are saying it's very confusing. So you have YR, then you have YR. So you have YR, and you have YR. So it's the same thing. So this is how we got it. So after that, then you would do the what? Do the crossing. So once you do the crossing, you will get the what? Multiple result, as you can see. So and from here, you would look at how many how many yellow and small do you have? We have this 1, 2, 3, 4, 5, 6, 7, 8, 9. So anytime, how do you discover yellow and smooth? Whenever you find one uppercase Y and one uppercase R, that means it's yellow and smooth. Even if you find one uppercase, so you can even have a double uppercase and double uppercase is the same. So you have one uppercase and one lowercase, one uppercase and one lowercase, it's going to be yellow and smooth. So when is the yellow and wrinkled? If you have one uppercase Y and you have double lowercases R, it's going to be wrinkled. So we would never be wrinkled if you have one uppercase R. So it will never be what it's never going to be yellow if you have one uppercase. So the meaning is once you have one dominant, it's going to be yellow for Y. And once you have one dominant for R, it's going to be smooth. It only becomes wrinkled when you have both recessive, and it's only going to be green when you have both recessive Y. All right, so this is how we got the ratio 9 to 3 to 3 to 1. So the hybrid cross in the inheritance of gene can be compared to the probability of flipping a coin. So if you flip a coin, there is a probability of having something or not having something. So the actual data might not perfectly match the predicted ratio, but with Mendel's result, there will be something close to it. And the amount of data you use or the amount of time you do the experiment will determine if you will get better result or not. So this is exactly what we have to cover. So there are some questions here, which is if a guinea pig has so if a guinea pig is got the if a guinea pig is got the it's got this, a guinea pig has this, and it's crossed with a white one. The white one has so the white one has this. So let's do it again. So we have a guinea pig. We have a guinea pig with a, sorry, mine. So the answer to this is just that when you cross one homozygous recessive, when heterozygous dominant, so you would have ratio of 1 to 1. So the answer would be this. So which gamete can not form from an individual that has a genotype? So which one can you not find from this? So is it this? Is it this? It is this. Based on what I did to you before, so we can find this and this. We can find this and this, but we will never find this because there's no double R in it. So, and a cat with a curled ears was crossed with a cat with a non-curled hair. All the kittens were born from the cross had not curled because the F1 generation would always be like that. So the traits will be hidden in one, and it's gonna be obvious in the F2 generation. So when the offsprings were being crossed with each other in the F2 generation, we had ratio 3 to 1. So it says which conclusion can be made? It can be made that what it is a recessive trait. It can be made that what the curd, so it can be made under what, because it says all the kitten born from the cross had non-curd. So the non-curd is dominant, so the curd is recessive, which describe an individual that is represented by heterozygous B in the Mendelian cross. Heterozygous, I already mentioned the answer. So this marks the end of Unit 1. Thank you very much for your attention.